Hello, everybody. This is Thomas Sir, CEO of Kinexum and EVP of the not-for-profit Kitalis Institute, which is co-sponsoring this webinar. The title is The Role of Biological Clocks and Other Biomarkers of Aging in Regulatory Development. There are a couple of housekeeping points before we begin. First, we are muting folks as they come in to preserve the quality of the sound. Please be attentive to keeping your mute on because this is a large meeting and it'll be disturbing to hear your neighbor's dog bark, etc. Second, because of the number of attendees, we can't really do a radio call-in show format, recognizing members who may want to ask questions, but rather, if you have a question along the way, please type it into the chat box, and the panelists will try to get to as many as they have time for. We are recording this webinar. A YouTube video of the panel presentation and the Q&A and a PDF version of the slides will be posted to the Kinexum website within a couple of business days. Registrants will receive links by email when these materials are available. Fourth, anyone interested in today's webinar will be interested in our Targeting Metaobesity 2020 conference, which will be held virtually this year from October 12 to October 15. Most of today's panel, along with other recognizable names, including Victor Zhao, Nir Barzilai, Brian Kennedy, Gordon Lithgow, Matt Kaberlein, Vadim Gladyshev, Aubrey de Grey, and Vittorio Sebastiano, and their peers in chronic diseases, government regulation, policy law, industry, capital markets, and other stakeholders will gather to seek ways to accelerate the translation of emerging geroscience into material accessible gains in public health. There is a free all-access basic pass option, and new this year will be a day 4 emerging company showcase on October 15th. So please check it out. Register at metaobesity2020.com. So the idea for this webinar arose out of an email communications I had with Steve Horvath last spring about the accelerating advances in the field of biological clocks and other biomarkers of aging, driven by advances in molecular, cellular systems biology, big data, AI, and a growing body of empirical findings. What we thought was needed, but we did not see, was attention to how to facilitate and accelerate the conversion of these continuously improving biomarkers into surrogate markers and registrable endpoints for regulatory development of therapeutic interventions. Just to frame some basic concepts, the FDA approves interventions on the basis of clinical benefit, like longer survival from a disease due to a treatment compared to control. Biomarkers are a biometric, a laboratory measure, a physical sign, or similar, that suggests a patient is improving, but the FDA is going to require evidence, validation that those metrics reflect and predict tangible clinical benefit, not just improving scores. At our Metaobesity 2019 conference last October, CEDAR Director Janet Woodcock cautioned the field from an overabundance of exuberant expectations, those are my words, not hers, that improving biomarkers would automatically, so to speak, be accepted by regulators as surrogate markers for regulatory approval. And indeed, as we consider commonplace surrogate markers like blood pressure, cholesterol, A1C, or viral load, it took decades of emerging scientific and medical consensus for them to become accepted as surrogate markers and registrable endpoints. Recognizing this, the question Steve and I discussed, which has resulted in today's webinar, is what can the geroscience research community do, if anything, to facilitate and accelerate the acceptance of biological clocks and other biomarkers of aging by regulators in their assessment of marketing approval applications? So next slide, DJ. Today's panel features some of the leading experts in the space to consider this question. Steve Horvath of UCLA is the discoverer of the DNA methylation epigenetic clock. Morgan Levine of Yale is the developer of the index biological clock offered by Elysium to support sales of their basis NAD+ supplements. Polina Mamoshina is chief scientist at Deep Longevity, is formerly a subsidiary of AI leader in silico medicine and recently an acquired subsidiary of Hong Kong public company Regent Pacific in the business of commercializing biological clocks. Luigi Ferrucci is scientific director of the National Institute on Aging of the National Institutes of Health and a widely recognized expert in biomarkers of aging and longitudinal aging studies. In addition to these geroscience researchers, we have Dr. Bob Temple, longtime preeminent thought leader at FDA. So here's how it's going to work. Each of the panelists will start with some remarks, starting with Bob, then Steve, then Morgan, then Polina, then Luigi. Then we'll have an intra-panel discussion. Then we'll open up to Q&A from the audience, taking questions from the chat column. So Bob, please take the mic.
Okay. Well, look, Tom has already said some of the things I'm going to say, but I will elaborate on them a little bit. And at the outset, you need to understand that the aging biomarker territory is completely unfamiliar to me. So I'm speaking generally and not specifically about these items. But what I understand is that there's a hope that at least eventually, if we can modify the markers associated with aging, or perhaps more rapid aging, we could change many aspects of aging, including cardiovascular effects, mental effects, muscular effects, and many more, with a single treatment that works on all of those things. And it would be directed at aging, not at the specific disease, the way, say, statins are directed at LDL abnormalities, antiplatelet drugs are directed at cardiovascular abnormalities, et cetera, et cetera, et cetera. So the hope, I think, is that there could be effects on a range of markers, not clinical outcomes, that could eventually be accepted as surrogates endpoints and support drug approval. The way we now use LDL cholesterol. You don't have to show a reduction in heart attacks. You just show that you change the cholesterol or blood pressure or hemoglobin A1C, and there are various other things. But as Tom said, those didn't come right away. There were huge, long debates, and the acceptance of obvious things now, things we now take to consider obvious anyway, LDL blood pressure and hemoglobin A1C, was a very long process. There was, it's interesting, I was in medical school about the time, something called the New York School in the 1960s that totally opposed use of blood pressure lowering because they thought it would lead to strokes and heart attacks and stuff like that, until the VA studies showed that that was not true and that in fact there was a benefit. Interestingly, there was a book on why LDL was a stupid endpoint, published just before the 4S and Waskop study showed that it was effective at preventing cardiovascular outcomes. So there was tremendous controversy, and there's always been fuss about hemoglobin A1C. So that sort of thing and other endpoints that I'll get to that didn't turn out to be beneficial means that we always want to see, pretty much always, there could be exceptions, an effect of a plausible surrogate endpoint and show that it actually leads to a well-defined clinical benefit. At that point, it can indeed become a validated surrogate and be used alone as a basis for approval. So what kinds of surrogates are we talking about? Most of them are an effect on what we think is the cause of the disease, and some of the aging ones probably are in that category. So that includes microorganisms. You know, to make a microorganism go away, it's pretty plausible that you make the infectious disease go away. Changes in blood pressure, elevated blood pressure, LDL, and hemoglobin A1C are well associated with bad outcomes, so they're plausible. Shrinking a tumor is plausible. Doesn't always lead to the effect you want, but it's plausible. Reversing hypokalemia or something like that. So those are sort of standard ones. You make the abnormality go away. And then sometimes you try to actually measure restored function. For example, people like the idea that improving ejection fraction in patients with heart failure might be a credible endpoint. Of course, there have been drugs that did that didn't have favorable outcomes, which is why everybody gets nervous and still wants to see the actual outcome. So, as I said, even for surrogates we buy, there are some inconsistencies. For example, you might have thought that what a statin does is open up the vessel that's obstructed by that's obstructed by the high cholesterol and stuff like that. In fact, though, the effect of statins is much more rapid than the effect on the vascular system. And that's because we probably don't understand what it works. It really probably changes the surface area very rapidly rather than the total volume of obstruction. I don't think everybody knew that, but there it is. And then we have a drug called torcetrapib that raised the HDL, which means it had to be a good thing because that high HDL is known to be good for you, but it worsened survival and coronary artery disease. And then there's been a whole series of heart failure drugs that had effects on cardiac output and cardiac function that actually impaired survival. So that could be because any drug not only affects the surrogate you're interested in, but may well have other effects that you have to know about, and you're not always able to anticipate them. It's interesting, though, that once we do have a surrogate, we don't ask for clinical studies anymore because they'd sort of be unethical. You can't really do them. And it remains possible that some drugs that affect the surrogate might not have the effect you want, but there's no good way to test for that. So you're sort of stuck with the accepted surrogate. So many of these matters are discussed in something called BEST, the Biomarker Endpoints and Other Tools group, which is a joint operation with FDA and the BEST group. A validated surrogate endpoint is one that is supported by clear mechanistic rationale and strong evidence from clinical studies that the effect on the surrogate endpoint has a clinical benefit. That's a validated surrogate, and those can be used to support approval, and I've already named some of the ones that we're talking about. The surrogate is almost always a biomarker: hemoglobin A1C, HIV, RNA, LDL, blood pressure, and there are some safety surrogates too. We worry about Hy's law, that's elevated transaminase, accompanied by elevated bilirubin, as a measure of whether liver injury is going to be really dangerous, and there are others like that. And what's a biomarker? A biomarker is a defined characteristic measured as an indicator of either normal biological function or biological responses to an exposure or intervention. And it includes all kinds of things, and it can help you define. One of the things everybody hopes for is that it's going to be able to measure effectiveness, but it can do other things that are very, very important. It can identify people who are susceptible or at higher risk. That allows you to choose them to put them in a trial. It can be the diagnostic marker. It tells you they have the disease, which is very important for knowing who to put into study. It can be used to monitor the effect of the drug. Even if you don't believe it tells you about the effect of the drug, it can help you choose the dose. You can have biomarkers that are prognostic, that predict who's going to do badly. You can have monitors that are predictive, that is, they seem to indicate who's going to respond, and things like that. So they're all very, very useful. As I said, biomarkers have many uses. One is a surrogate, but it's very important that they can help you do better at patient selection. And all the things we're talking about here for aging, you could use people with the more extreme versions of those markers to put into your trials because they're likely to do worse if you believe that. So it can help tremendously on that. And okay, and so as I said, the identification of biomarkers as mechanistic, predictive, prognostic, that's great. And for now, however, even if you think you've got those, we're probably going to still want to see that the drugs really do what they hope to do. And everybody's looking closely at the TAME study, which uses a very interesting endpoint, as probably everybody knows. It uses an endpoint that includes cardiovascular events, cancer, dementia, mortality, and a bunch of other stuff. It has many functional measures. It'll be very interesting if that comes up right. But just as an example, suppose the total result is driven mostly by cardiovascular events. Will you think you now know something about its use, about aging biomarkers? I don't know. We'll see, but it'll be very good to look, and we'll get to look at all the little endpoints within there and form new hypotheses and so on. And that's the end of my intro.
Thank you, Bob. We're going to save questions until the end. So can we go now to pass the mic to Steve Horvath from UCLA, please? Thank you.
Hello, everybody. I have been asked to give a short history of epigenetic clocks. And naturally, I have to be succinct and cannot mention everything, but maybe we'll start with the next slide. It all started probably in 1967. There was a Russian researcher called Berdyshev, and he showed that in the pink salmon, global methylation levels decrease with age. Global meaning the average in certain ways across all the millions of locations in the DNA. Next slide, please. In subsequent decades, the 90s and up to 2010, there was a very detailed characterization of genomic regions that gain methylation or lose methylation. And many of these studies were motivated by cancer applications, but also there were many aging researchers who characterized aging effects. And again, this would be a 2-hour presentation to describe that CPG islands gain methylation and so on. 2008 saw the introduction of a new technological platform, the so-called Illumina Infinium Methylation Array. And many epidemiologists welcomed this array platform because it allowed you to measure cytosines in large epidemiological studies. Next slide, please. So how did I get into the field? I had a collaborator at UCLA who was interested in studying epigenetics of homosexuality. And he had assembled a very nice data set of saliva samples from homosexuals and controls. And we simply didn't find any significant result. Nothing was publishable. But I, of course, was very interested in aging effects. And I realized that one can build these so-called multivariate age estimators based on multiple cytosines combined. And this was the first epigenetic clock for human saliva samples. Now that was a cute application. You spit in a cup, you have a saliva sample, you can measure somebody's age. And so it was cute, but nobody could realize that this has more promise. Next slide, please. And this brings me to a discussion of the definitions of the term epigenetic clock, because this term is loosely used in different papers. I call it old definitions are that an epigenetic clock is an unknown biological process that causes aging by affecting the epigenome. So it's a rather vague definition, similar to the definition biological clock. And other people use the term epigenetic clock as being synonymous with age-related changes in the epigenome, or even more precisely methylome, But in this talk, I use the new and precise technical definition. For me, an epigenetic clock is a multivariate estimator of age or mortality risk. In essence, there's a formula, a mathematical formula. Next slide, please. 2012 and 13 saw the publication of very interesting blood-based clocks. There was an Italian team that showed that a single cytosine in the so-called ELOF-L2 gene exhibits remarkably high correlation with chronologic age, above 0.9. And similarly, Hannum, Eidecker, and Zhang published the so-called Hannum clock based on 71 CPGs. Again, a multivariate estimator of age. Next slide, please. 2013 saw the introduction of a new type of clock. This is referred to as a pan-tissue clock. Why? Because it applies to all cell types of the human body. And as we discuss later different biomarkers, I want you to remember, you should always ask yourself, what biological material does the biomarker apply to? Some biomarkers require blood plasma, most require blood, but really the remarkable thing about epigenetic biomarkers of aging is that they truly, you can define them so that they apply to all tissues and organs. And what do I mean by it? There's one formula, one mathematical formula for estimating age. Moving on to the next. 2015 saw the publication of several large meta-analyses that leveraged existing epidemiological studies where people had generated data using the Illumina methylation array. And so several groups showed that after adjusting your biomarkers for age, sex, BMI, smoking, all these confounders, these biomarkers still lend themselves for predicting lifespan. Why? Because these studies had collected blood samples in the 1990s, for example, and there was follow-up information on everybody. So we knew how long somebody ended up living, what they died from, and so we could show that these biomarkers are predictive of lifespan. Next slide, please. 2017 saw the publication of several mouse epigenetic clocks by several teams. And these clocks were remarkable in that they showed that well-known anti-aging interventions slow epigenetic aging in the mouse. Most of you will be familiar that caloric restriction has great benefits, especially in rodents. And sure enough, caloric restriction is associated with slower epigenetic aging. And that's a very robust effect. You can see it with any mouse clock that you can come up with. There's another gold standard intervention, the so-called growth hormone receptor knockout mouse model. In other words, dwarf mice have been shown to live longer lives. And sure enough, this effect is also extremely robust. Most mouse clocks will show a profound epigenetic reduction in age. Moving to the next slide. 2018 saw the introduction of the so-called second generation clocks. I briefly want to go back. What is the first generation epigenetic clocks? The first generation epigenetic clocks allow you to estimate chronologic age. The second generation clocks should really be interpreted as estimators of mortality and morbidity risk. There's the pheno-age clock, Morgan Levine and Luigi introduced it, but also Grim age from Ake Lu. And these clocks, especially Grim age, is really not an age estimator, it's a mortality risk estimator. Now, you can publish anything, and it's important that others validate it. So if we go to the next slide, I want to discuss some evidence for Grim age. By now, there are over a dozen papers by independent groups, independent in the sense of they use these biomarkers in different cohorts, ranging from Scotland, various European cohorts, all over the world, really. And to validate these claims that Grim age, for example, applied to a blood sample predicts a lifespan or morbidity risk or time to heart disease, and so on. So there's no debate that it validates in an epidemiological sense, in a cross-sectional study. And more recently, Riccardo Marioni showed that polygenic risk scores for Grim age are strongly associated with parental longevity. And so there's genetic evidence that these risk factors predict lifespan. And more recently, and that's kind of the cutting edge of epidemiological studies, is to associate rate of change in grim age or any of these biomarkers with hazard of death. If we ever want to convince the FDA and others and ourselves that these biomarkers are useful surrogate markers of endpoints, we need to establish causality. And that one tool in that are, of course, longitudinal studies and to a lesser extent, genetic studies. Next slide, please. 2019 saw the publication of what I call a futuristic study, unexpected. So Greg Fahey and Bobby Brook had developed an exciting cocktail of substances to regenerate the human thymus. This was a cocktail that used metformin, growth hormone, DHEA, and other substances. And there was a very interesting side effect, epigenetic rejuvenation. I call this study futuristic because I thought these kinds of trials will be carried out 30 years later, you know, but there is a limitation. It's a phase 1 trial, not meant to show efficacy. It was meant to show safety. But next slide, please. And here I show you the results for the grim age clock in that trial. So people started at baseline month 0, and they took the treatment for 12 months. And I think you see that after 12 months, there was a grim age reduction of 2.5 years in these people. Mentioning again, it's phase 1 trial, so I'm very happy that Bobby Brook and Greg Fahey are now carrying out an expanded pilot clinical study where they will enroll about 100 people to validate these original claims. Next slide, please. Now it's 2020. What's new? Here's what I call the third generation of epigenetic clocks. I refer to them as multi-mammalian species clocks. Here I show you a clock, a single mathematical formula, but it applies to rats and to humans at the same time. The left panel shows you in green human samples. The orange dots on the left are rat tissue samples. And you see good mixing. Now what's on the x-axis? That's relative age. Relative age is age divided by the maximum lifespan of the species. So for example, in a human, the maximum observed lifespan is 122. So if you're 61 years old, your relative age is 0.5. Next slide, please. We have now developed these dual species epigenetic clocks for many mammalian species. I just mentioned that if you own a cat and you send us a blood sample from your cat and from yourself, I'll measure your age and that of your cat using the same mathematical formula. Moving to the next slide. Same for elephants, except I don't think you own an elephant. Moving to the next slide. So we are now working on so-called universal mammalian clocks that measure chronologic age in over 100 mammalian species. Moving to the next slide. So here I show you a glimpse. Each dot is a different tissue samples. In the upper panel, you see a test set of 3,000 tissues, many different mammalian species. But you can already see that these universal clocks are actually remarkably accurate. And what does that mean? If you have a species and let's say you study bats or any other species and that you catch in the wild, you don't know the age of that animal. We can estimate the age of these animals. So by the way, why do I do these studies? Because I believe that aging processes are conserved across mammalian species. So if you have a clock that applies to all species, arguably it captures important biology, that's a universal pathways causing aging in many different species. That's really the motivation. Moving to the next slide. So what will happen in the future? What will happen in 2021 and downstream? And many people, fortunately, work right now on this important problem of mechanistic characterizing these age-related epigenetic processes. We simply need to understand the causes and consequences of methylation in general, but also epigenetic clocks in particular. There's also a need to further develop and optimize epigenetic biomarkers, and of course, many complementary biomarkers for human clinical trials. I, of course, focus here on epigenetic clocks, but I'm very cognizant of the fact that we need additional biomarkers in order to measure the many facets of aging. Next slide. So just to remind you, by now there are many human clocks. They do differ in predictive accuracy for mortality. For example, the pan-tissue clock is not a good predictor of mortality risk. The Grimm age clock would be a wonderful predictor of mortality risk. DNA methylation age will not replace standard clinical measures. Bob Temple just discussed the very important biomarkers from LDL, HDL, and so on, blood pressure. They will never be replaced by methylation biomarkers. As you design your study, you really need to think very carefully about what tissue or cell type you want to study. Some anti-aging effects manifest themselves by reversing blood-based clocks, but others don't. Other anti-aging interventions reverse epigenetic age in buccal epithelial cells. So for example, hormone treatment, female hormone treatment seems to reverse epigenetic age in buccal cells. And as people develop these biomarkers, they should also consider, for example, urine sample. You can extract DNA from urine, and it's very predictive of age. Adipose tissue. Luigi Ferrucci even manages to collect muscle tissue, so he can tell you all about it. And I'll stop with the acknowledgement slide. Thank you.
Super, thank you. Thank you, Steve. I said we weren't going to do questions first, but I see a couple questions coming through. Steve, how would you characterize the clock or clocks or your clocks that you talked about with the telomerase clock? Do you have any comments on that?
Yeah, sure. I call it the telomere clock. I hope this is a telomere length clock. I hope that's the same clock we're talking about. Telomere length is a beautiful biomarker because there's a very detailed mechanistic understanding. We know the enzymes and so on, and that makes it and also telomeres are part of every cell, and so there are age-related changes in most cells. So from the outset, telomere length is the ultimate and beautiful biomarker of aging. I certainly loved it when I first came across it. But then, unfortunately, there is a harsh reality and a great disappointment to the entire aging field. And this is that telomere length is actually, at best, very weakly predictive of outcomes we care about. It's an exceedingly bad predictor of lifespan. It even is a bad estimator of chronologic age. If you had a telomere length, you could never estimate chronologic age in people. I also want to briefly mention an anti-aging intervention according to telomere length, which is older fathers. So if you wanted to cure aging in the population according to telomere length studies, you would tell men they are only allowed to father a child after age 16. Okay, so So and also so anyways, unfortunately, telomere length has not proven itself in large epidemiological studies.
Steve, I'm slapping my hand. Thank you. There are other interesting questions popping up, but I want to make sure that we get all of the comments out. So if we can switch, throw the mic over to Morgan. Please take the mic.
All right, perfect. Thank you so much. And I think, Steve gave a really great introduction to a lot of the stuff I'll be discussing. I'm going to show very little data, but mostly talk about the things that I think are important when we're developing some of these biological age markers or epigenetic clocks that could potentially be used as outcomes in various clinical trials. Oh, so next slide. So as was kind of covered already, biomarkers of aging are really useful proxies that can be used in a number of different applications. So number one, which we're mostly talking about today, is in clinical trial endpoints. So rather than having to do a clinical trial that lasts 10, 20, potentially 50 years, if we actually did have good proxies for the biological aging of some tissue or cell type, we could use these to represent what we think the mortality or morbidity effect might be. Another application is just in better understanding of the basic biology of aging. And then finally, in risk stratification. So looking at populations, identifying at-risk people, whether that's for recruitment into different interventions or just better monitoring. So the goal of these biomarkers of aging are first, most simply just to differentiate someone who's 20 years old from someone who's 8-year-old. But probably more importantly is among people who are the same chronological age, can we differentiate people who are accelerated agers versus those who are decelerated agers? So people who are aging faster than we would expect. And then finally is to actually look at modifiability. So can we actually intervene in the aging process and actually assess whether some specific intervention is actually effective in slowing the aging process. So next. So when thinking about actually measuring biological age for a clinical trial or basic research, there's actually a number of different, what we might call biological levels at which you could measure biological age. So we actually think that aging probably starts at some of these lowest levels of biological organization, so at the molecule level, and actually propagates up. And really what we see at the organismal level is kind of the emergence of the changes that are happening at these lower levels. So technically, you could actually measure biological age at any of these levels, and which one you pick actually might depend on what the question you're asking is. So next. So in developing biomarkers, I think I kind of want to focus on 4 major goals for this talk. So first, validity, reliability, modifiability, and finally, mechanism-focused biomarkers. Next. So starting with validity. So what does this mean? Is one big issue in actually developing biomarkers of aging is that aging is a latent concept. It's actually something we can't observe. And so anything we do is not going to have a ground truth for which we can evaluate it. So Steve already did a really great job at kind of discussing the first generation versus second generation epigenetic clocks. So again, these first generation clocks and a lot of biomarkers of aging, even beyond the ones developed in using DNA methylation, are developed as what we call chronological age predictors. So you get thousands to potentially hundreds of thousands of variables. You do either some machine learning or deep learning technique to see if you can actually predict the chronological age of a sample. So one issue with these types of clocks in actually applying them either for clinical trial use or for monitoring health and aging is that in training these, the algorithm may not prioritize the most critical biomarkers for health outcomes, for mortality risk. And that was pretty well demonstrated actually in the epigenetic clocks, where we found that the ones that are actually trained not on chronological age ended up being better predictors of the outcomes that we're most interested in. So some reasons why this might happen is that you actually might be adjusting out some of the things that create differences, biological differences among people the same age. So obviously, if the algorithm is trying to get the best predictor of chronological age, it might actually reverse weight some of the things that differentiate people of the same age. Another issue is mortality selection. So if you're getting very accurate age predictions for people who are 80, 90, or 100, those are probably not biomarkers that actually have an impact on mortality, because people who are 90 or 100 are not some random subset of the original birth cohort. They're actually the most resilient selective people. So they actually should be predicted younger than they are. And if you're getting good age predictions in them, you're probably not measuring things that actually have an impact on mortality. So this is kind of where we came in with what we call these second generation measures. So can we actually measure things or actually predict things like phenotypic aging or some proxy for biological aging and develop biomarkers for them? So we think that by doing this, we'll actually be capturing more important information. But there are a few drawbacks. So number one, it's a lot easier to get a huge sample for training if your outcomes age. Age is really easy to measure. So most of the data available for making these second generation clocks is going to be a lot smaller. And then the other issue is that it's not entirely clear what our outcome actually should be, and whether we should combine a number of different outcomes, whether we should just use things like mortality, which might prioritize things important for cardiovascular disease and cancer, or whether we should combine a bunch of different outcomes to make better kind of phenotypic age predictors. Next. So the second important thing in thinking about designing these biomarkers of aging, whether for clinical trials or consumer use, is reliability. So basically, if I take multiple samples from the same person, whether it be on the same day, whether it be a week later, do I get the same answer at an N of 1 level? So this is something I work as a bioinformatics consultant with Elysium Health, and they recently put out an epigenetic biological age predictor called Index for consumers. And it was really important for us to make sure that we were giving someone a reliable answer when we were measuring their epigenetic age. So unfortunately, when we actually looked at the existing epigenetic clocks, we found that actually at the N of 1 level, they're quite unreliable. So this chart is based on 9 technical replicates taken from each person. We had 8 subjects in this. And this shows a number of different epigenetic clocks that have been developed. This is all done in either blood or saliva. And what you can see is actually you don't get that good agreement in terms of the age that these samples are predicted at. However, we've developed different computational statistical ways around this in developing Index that have actually really improved the reliability, which we think is going to be very important both for consumer markets, but also in clinical trials. If you're saying that your intervention is decreasing biological age by 1 or 2 years, that should hopefully be a bigger number than your error rate. So this is really important in that regard. Next. So the other important thing is modifiability. So can we actually develop biomarkers that are modifiable? And this is showing some preliminary data. I'm not going to go into what this intervention is because it's unpublished, but basically, can you apply some intervention and see a change in whatever biological age measure that you're measuring? And the important thing that I think is still an unanswered question in the field is you can have an intervention, you might see some change in your biomarker of interest, but actually does that impact downstream morbidity and mortality risk? So I think that's really where the field is, and I think it'll be really critical for some of these studies like TAME, where we actually have an intervention linked with some of these biomarkers of aging, and then also following up and looking at morbidity and mortality risk in these populations to actually see whether these things are all linked up and actually try and understand better. Steve mentioned causality in terms of the biomarkers. And next. And finally, kind of as a last thing, I think we really need to go back and focus a little bit more on kind of mechanism. What are we actually capturing in these biomarkers? It's not entirely clear, particularly with the epigenetic clocks, what actually drives the changes in DNA methylation that are captured by them. And in fact, whatever your intervention is, actually you might want to choose a different biomarker, also going back to those different levels of biological organization based on what you're trying to target. And the only way to do that is to actually know mechanistically what we're capturing. So my lab has been doing a lot of work where we're actually, in some ways, dissecting the various epigenetic clocks, and what we're finding is that they're actually composites of a lot of different mechanistic changes that you kind of add up to get your overall clock score. However, some clocks are capturing more of one type of mechanism than the other, and it's really important to know what these different biomarkers are actually proxying. And this will also help us in terms of identifying interventions that actually might impact these measures as well. So next. And with that, I just want to acknowledge first and foremost the people in my lab who have been doing a lot of this work, also my funding, and then finally the leadership team at Elysium Health, who I've worked really closely with in developing index, which we're really excited about. And thank you so much.
Thank you, Morgan. We do need to zip along. So EJ, would you please queue up Polina's slides? And Polina, you have the mic.
Thank you. So after those great introductions made by Steve and Morgan, I just want to more focus on the fact that these models, those aging clocks that we are all developing, are computational models in the beginning. And they provide us with the great tools for simulations without actually going to wet lab experiments. And we can even run in silico clinical trials with them. So I think that's what is the most important for them. So if you go to next slide, I just want to do a brief disclaimer that I'm a chief scientist at Deep Longevity. It's a company that focuses on developing deep aging clocks, but we are trying to publish as much as possible in the peer-reviewed journals, have multiple publications, and slides today that I will show you will be covering mainly those. We are founded by Professor Alex Zhavankov, who is also original inventor of those so-called deep aging clocks. We call them deep because we use deep learning to develop them. And if we go to next slide, I just want, again, to emphasize that there is a lot of aging clocks available right now. And it's, let's say, started in 2013 by the publication of Professor Steve Horvath on epigenetic aging clocks. Yet all of them were developed using so-called shallow machine learning. So it's a really standard linear regression models that are mapping the changes in epigenetic profiles to chronological age and predicting it. And if we go to next slide, while we have a completely new technology available to us, so deep learning, and it's changing a lot of industries, including image recognition, voice recognition. We have self-driving cars right now. While most of those ideas in artificial intelligence or deep learning, they came from 90s and 60s, it's only become doable in 2014 with deep learning revolution in imaging with the ImageNet competition. And most of today hypes around AI and deep learning in general is mainly driven by deep learning and actual advances in deep learning and reinforcement learning. And we believe that in 2019, deep learning started to revolutionize not only imaging and voice recognition industries, but also healthcare. And if we go to next slide, we can see why it's happening right now. Why we had the technology from the early beginning, but why it's happening right now? Because we actually have algorithms that allow us to train them. Before it was only theory. Now we can do this computationally. We have computing power. So we have computers, supercomputers, we have GPU computing, so we can actually train those algorithms, and we have big data. We have millions of samples, so they are exception data hungry. We have millions of samples to train them on. And if we go to next slide, we can actually see that FDA as an agency already recognized that. And since 2017, granted 14 approvals for purely AI-based tools. So purely those technologies were developed using deep neural networks. And for most of them, those were fast-track approval, just based on their superior accuracy compared to tools that are available out there. And it was all started in 2017 with Arteris Cardio and their MRI segmentation tool. And one of the recent approval is for Apple atrial fibrillation detection that is actually available right now in Apple Watch. So if you have Apple Watch, you already have an AI-based tool that can predict whether you will have or having atrial fibrillation. So if we go to next slide, we will see that there are a lot of developments in aging clocks, deep aging clocks. So a lot of development with this new technology started in 2016. And if we compare deep learning with standard machine learning techniques, actually there are more clocks available here with a really completely different data type. And for some of those clocks, especially for image-based clock, like X-ray, MRI-based, or facial images even, it's not even available to, it's not even possible to predict age without deep learning. So if we go to next slide, I want to just show you what is the difference between a really standard linear regression model and a deep model. In a linear regression model, you're just mapping your markers to the age or mortality, wherever, depending on what is your outcome. But in deep model, you actually have hidden layers. And in this way, you are able to capture really highly nonlinear dependencies in data and your outcome. If we compare them by accuracy, we will see that the deep models, just based on their architecture, are way superior compared to shallow models. If we go to next slide, I just want to draw your attention that I don't personally believe that we will be able to construct a universal aging clock. And this is an amazing study that was done by Professor Dandelsky and his colleagues in 2018, published in 2018, where he showed that if we analyze biological age of individual the same chronological age using different aging clocks and different biomarkers of aging, you will see there is low or no correlation between them. And while those individuals are presumed to be healthy, and there is a low correlation from, let's say, mathematical point of view between even epigenetic aging clocks. So measures don't agree between each other. At the same time, if we analyze the longitudinal data, we will see that clocks are ticking in the right direction. So people without interventions, they get older, not only chronologically, but also according to those measures. So they are working, just they measure different things. They measure different aging processes. And on the next slide, you will see that that's why we are trying not to focus on a single data type, but trying to build a really diverse portfolio of aging clocks because we want to capture completely different processes of aging. And that's why we work with data ranging from simple clinical blood tests, like blood biochemistry and cell counts, going to microbiome data. So if we go to next slide, that will be the first marker I want to show you. I will be really brief here, but just if I analyze data itself, blood markers and the changes in their levels with aging in healthy people. In healthy people, we would assume that of course there will be variation in their biological age, but still, because we expect them to have similar life expectancy, if they have similar lifestyle, they are healthy and so on. We will still assume that the chronological age should be close to their biological age. Of course, there should be variation, but it still should be close. So if we analyze the blood markers levels in healthy people, we will see that those common criminals like albumin cholesterol, they change with age. Yes, there is a strong correlation, I mean, some correlation, some trend. So now albumin increase with age, cholesterol, or I mean, albumin decrease with age, cholesterol increase with age, glucose increase with age. But it's not like you can actually predict a number of assign a biological age based on those values. That's why you need to have a model, and that's why you actually have to have a model that capture really highly nonlinear dependencies between changes in blood markers and in age. And we did this with the deep neural networks, actually with an ensemble of those, but it's not important. But what was important that with the simple blood parameters, clinical blood parameters, we were able to predict age of healthy individuals with error of 5.5. So if we go to next slide, we also will see that, yes, we can predict age of healthy people, but when we analyze different lifestyles or when we analyze their mortality, we will see that there is association with this age acceleration and different lifestyles. For example, tobacco smokers are predicted twice as old compared to their non-smoking peers. The same for mortality. If we analyze 5-year mortality, we will see that people who are predicted older, they have higher hazard ratio, and so lower life expectancy and the other way around. But if we go to next slide, we'll see that blood biochemistry is not the only data type we can work with. We can do the same for microbiome. Again, with a reasonably good accuracy predicting age. Also, we can see that people with diabetes are showing signs of accelerated aging. So they're predicted older compared to healthy peers, same chronological age. And if we go to next slide, we actually go into probably the most important slide of my presentation today, is that not only we can predict age with those models, but also we can evaluate how the input parameters are influenced at this age. So this particular example we did for microbiome. What we did here, we changed levels of the or representation of those microbiome species in the profiles, and then we saw whether the predicted age was bigger compared to original sample or slower. In this way, we were trying to analyze whether the changes in those species or abundance, those species abundance is actually associated with the increased or decreased age. In this way, we assign them SENA-negative when they decrease age and SENA-positive status. And we were able to classify them in a way. But that's actually a really powerful tool if we want to experiment with interventions right away, because there is no need to go for clinical trials right away with this. We can just simply experiment with input parameters, trying to identify what kind of parameter are the most important one, and what will happen with the biological age predicted by all those clocks once we change them and in each direction. So we can also experiment with the combination dose and so on and so forth. So if we go to my next slide, that's a summary slide that shows that, yes, of course, we have a lot of aging clocks available, and definitely there's a revolution happening right now in terms of algorithms in healthcare. And FDA recognized the potential because, again, most of those 14 AI-based tools that were approved, they were granted fast track approval. At the same time, those clocks are not only useful in predicting biological age, but also useful as computation tools when if we want to simulate outcomes. So it's compared to simple markers that are out there like cholesterol level or glycated hemoglobin level, it's something different. It's a computation tool. And this way we can use them to simulate effective interventions. So with that, again, I want to thank you for attention. And I want, again, thank organizer for having this meeting. So I think it's about time, and it probably will be a historical meeting if we can actually have this discussion with the FDA right now on aging clocks and how we implement them in clinical trials.
Thank you, Polina. Switching quickly to Luigi. EJ, if you could queue up Luigi's slides, please. Luigi, you have the mic.
Hi. Very nice meeting so far. You know, 3 really exciting and complementary presentation. And my duty here is to understand not how beautiful they are, but basically what we have not done yet, which is much more difficult thing to do from the perspective of somebody that really love the study of aging, such as myself. So if you get the next slide, I want to just talk about what we have done so far in medicine. What would be our paradigm? And our paradigm has been that disease are due to a combination of genetic predisposition, environmental risk factor that lead to pathology accumulation with clinical manifestation and functional consequence. And the diagnosis of disease, identification of a specific risk factor for that specific disease has been a paradigm for every type of discovery and treatment that we have done in medicine. So here I am simplifying, what are the risk factors for heart disease. And we know the smoking, alcohol consumption, high cholesterol, hypertension, diet, inactivity, obesity are all related to heart disease. So when you go to a cardiologist, it's going to tell you to stop smoking, exercise, and look at your cholesterol and diet. Next slide. However, we can see that, I think you have going too fast. Can you go to the previous one? Okay, so when you look at what's going on, you find that the smoking, in fact, is not only a risk factor of heart disease. Smoking affects cancer, increase the risk of injury, chronic lung disease, is associated with stroke, dementia, and diabetes that are all the major disease that occur with aging. And the next slide, in an attentive exam of the literature, we'll find that there's a complex network of interaction between these risk factors and many, many diseases. And then tell you that there is an underlying homeostatic and health mechanisms that drive the development of all these diseases. It's not by chance that all these diseases have as a major risk factor the aging process. The next slide. So we need to talk about how do we capture these essential process that is underneath the development of disease and the reduction of health expectancy in some individuals. And here I show you one of the biomarkers that geriatrician and gerontology have loved for more than 30 years, which is a simple one, walking speed. You can take somebody walking for 3 meters, measure their walking speed, and these are data from my Italian study, the Incanti study, showing that those that are walking at a speed of less than 0.8 meters per second have a risk of mortality over 6 years that is higher than those who have a diagnosis of malignant cancer. So it's an incredibly powerful biomarker. It's not something that you get in the blood, but it's something that is more predictive of that outcome than having a diagnosis by a doctor of malignant cancer. Okay, so that's one. And what's wrong with that? Why do we need something more? I'm gonna get to that, but give me the next slide. The next slide show you another risk factor that is incredibly important for aging, which is multimorbidity, which is by chance, the paradigm that is being used to create the outcome of the TAME study. The TAME study individual will be individual with one disease, and the outcome will be the delay in the development of the second disease. But as you can see, the number of multiple chronic disease increase with aging, and these are longitudinal data. And what is interesting in this slide is that one of the blood biomarker that we love the most, interleukin-6, is associated with accelerated increase in multimorbidity. Not only a change in IL-6 is affecting also the rate of accumulation of multimorbidity. Next slide show that if we look at inflammation, this is a very old paper showing that those with an IL-6 higher 2.5 picogram per milliliter have an increased risk of losing mobility over the next 4 years. So a powerful, powerful predictor, and yet, walking speed, multimorbidity, interleukin-6, and inflammation are not included in clinical evaluation and are not considered except in some very specific indication biomarker that can be acceptable for the evaluation of drug. of course, there are evaluation that the exception that we need to take into account. The monoclonal antibody that inhibit IL-1 beta that prevent cardiovascular disease is an excellent exception where we can show that the effect is higher for those that show more decline in health. So how do we overcome this, and how we go from the beautiful presentation that we have seen on these biomarkers that are based on omics, or also multi-omics and artificial intelligence, to something that is tangible. The next slide. The next slide, I tried to outline what do we need to do to go a step forward. What is the roadmap for going a step forward? The first thing we need is to provide strong evidence that biomarkers are associated with multiple independent health and functional outcomes in humans. If you only predict cardiovascular disease, you can't really think that this is a global biomarker of aging. You have to be able to predict multiple disease and conditions that are being associated with aging. Wrinkling of the skin, elasticity of the skin, sarcopenia. All these outcomes that are related to aging needs to be associated with these biomarkers. We need strong effect. We play with p-value so much as researchers, but as clinicians, we want strong effect size. And also we want these to be tested in real clinical population, because we know that the data from epidemiological study are often biased by survival and by participation. And then most of all, and I think that this will be even, if this is the only one that come across from my presentation, I'll be happy. We need longitudinal data. Cross-sectional data are really inadequate to say that a biomarker can be clinically useful. We need to say that certain biomarker predict future outcome. We need to demonstrate that change in that biomarker are associated with multiple different change of the aging phenotype, including disease. Example exists, and we need to show that in the long term they are associated with global health and longevity. And when we have demonstrated that association, then we can use them as surrogate marker. But we can't really say that a surrogate marker based on cross-sectional data. The last thing I want to say is that we need to carefully realize the biomarker can be both a measure of damage accumulation and resilience. It is possible that what we're measuring is just the reaction of our body against the damage accumulation. The change in those biomarkers will not be the way to go, but will be recognizing the mechanism, will be the most important thing we can say. One last thing I want to say is that if we start doing clinical trial, and we will have to do clinical trial in people that are relatively healthy, if we want to demonstrate that we improve lifespan, these interventions will have to be incredibly safe, because we're targeting people that are apparently healthy and no disease. And so that will be the difficult bar that we will have to demonstrate. The next slide. Okay. This slide, I want to just say that the need for longitudinal study is intrinsically related to what we know about aging. So we know that many of the damage accumulation that occur with aging is reduce or is offset by many biological mechanisms of compensation. You can have damage of DNA, and the DNA damage lead to repair. You may have misfolding of protein, and we have powerful chaperone. And so until you have a critical threshold of biological damage, you're not going to have phenotypic manifestation. That means that at the time when you're more effective in changing the biology of an individual, the individual will be healthy. And so we need to look longitudinally whether preventing those change of biological damage before the the phenotypic change or the functional change, such as the walking speed. At that level that we need to provide effectiveness. But also this slide serve me to show you something. If you look here, when we have functional aging, the declining walking speed, of course, those are powerful predictors of outcome and mortality. Because at that time, all the resilience strategies that this individual had have been exhausted. But it's too late. So we need to make a diagnosis before the functional manifestation occur, and that will be taken into account in the design of our clinical trial. Can I have the next slide, please? This is really my last slide. And I want everybody to think a little bit of how powerful this biomarker can be in the future. Each one of us have gone to the doctor, at the time that we're very healthy and asked to have a checkup. What's right, what's wrong with me? And my experience and the experience of many of my colleagues is that we go there, our cholesterol is fine, our blood pressure is fine, we eat well, we haven't gained weight, and so we are sent home with a ticket of being healthy. But we know that there is damage accumulation within our body. And so in our mind, am I in trouble? Is something wrong with me that at some point will show up? Is there something I can do about it? And what I'm doing the right thing to do? It's only if we can capture those biomarkers that we'll be able to do that. And this is another use of biomarker that is not for clinical trial, but a very, very important clinical use for the future. And I'll stop here and thank you for your attention.
Thank you, Luigi. Fascinating presentations. We could go all day or all semester in a course, I guess, to talk about these things. A little self-flagellation is in order. When we originally planned this, we were trying to leave at least half an hour for discussions and Q&A. There's some wonderful conversations going on in the chat. What we'll probably do is to capture the questions, pass it on to the panel, try to get some answers and distribute the answers. I think back to the focus of this webinar, which is how to facilitate the conversion of these biomarkers and clocks into surrogate markers and registrable endpoints. I think we can, if we're good if we can focus on at least two questions, and one of them is talking about biological clocks and the fact that they measure different things. One of the issues is, aging is not considered a disease currently, and a disease or condition, and does that need to be come about in order for biological clocks to have a role in therapeutic development? And the second would be, we've talked about a lot of different aspects. We understand that clinical validation, if we can come back with a successful TAME trial and talk to Bob and others at the FDA, we can have a conversation. But are there other things that the geroscience research community can focus on, divert 1% of their brain power to generating data, whether it's mechanism, causality, reproducibility, etc., that could help the FDA to come around to thinking about these biomarkers as something that can be relevant in their assessment of marketing approval applications. So let me start with a question generally to the, well, let me throw it open, I guess, to the panel. Bob, first dibs, what is your reaction to hearing some of the cutting edge research that was presented by experts in the field? Do you see it as a promising avenue for development of therapeutic interventions? Bob, you need to unmute yourself on the bottom left there.
Right,
Thank you.
I think it's all extremely important, but as I sort of said before, it seems most important in helping you design the trials that are going to prove that it really matters. And I had one point I wanted to make in response to the third from the last slide of Morgan's, where she was looking at change in bioage and control and intervention. It was quite striking to me that there was a large range of responses to the intervention. some of which hardly changed at all and some of which changed a lot. If you wanted to do a trial, one of the things to enrich it is to do a pre-screen for the people who have the biggest response and then randomize them. If you're right about what the intervention is doing, that will get you the people who are the hyper responders. And past experience shows that that's an overwhelmingly efficient way to show something. It still implies that you have to show something, which, I'm not sure I understood everything that was being talked about, but that still seems important. I mean, after all, we're talking about interventions that conceivably would be applied to the entire population. You better be sure you're right, and that there's nothing funny going on. But that kind of enrichment can make an enormous difference.
I can comment quickly on that. I mean, I absolutely agree, and I'm not going to go too much into that trial, but we were able to identify what distinguished the responders were, and it was pretty simple. The non-responders were people who started off in extremely good health. It wasn't necessarily just a regression towards the mean, so but you could really see that there probably is a floor effect on some people, that they're only going to improve so much. So I agree with Bob. If you can also use these biomarkers for pre-selection on who actually needs the intervention, I think.
Yeah, it makes another point. There's been some discussion that suggested that you want to get to people before they're damaged. Historically, that's not always true. Sometimes the more efficient trials take people who are already showing some damage and show that you can improve it. It's been true in every intervention we've talked about. Secondary prevention has always been done before primary prevention because you have more events to try to reduce. But it hasn't gotten in the way. People are not implacably damaged when they've had their first heart attack or stroke. And those populations have been the very ones where effectiveness has been shown first, historically.
Any other reactions from the panel? We've heard this interaction with the FDA. You know what's happening in the research program enterprise. What can we do to help meet the regulators' needs and interests?
Can I contribute? But just this is Luigi. I think that there's a way to go before we can use this as outcomes. But wouldn't it be nice to use the current clinical trial to understand how good of outcomes these measures are? Can we try to introduce those measures as parallel measures in some of the promising clinical trial? And by doing that, without having to do a new clinical trial, collect the data that we need really to demonstrate the importance and the robustness of these biomarkers. I think that a push in this direction will give us really a rapid demonstration, because this can be done in many, many clinical trials at the same time.
Yeah, I fully support that. There is an element of serendipity in science, and if we piggyback on existing clinical trials where people typically carry out blood draws, and if we could just divert a little bit of this biological material for various biomarkers, we open up true opportunities. And I remind you of my presentation where I described a clinical trial for thymus regeneration, but it really appears to have a pretty strong effect on epigenetic clocks. But I hope that these kinds of discoveries can be made with many interventions. And regarding the point, is aging a disease? We could have probably an entire workshop on it. There are philosophical answers, there are ethical answers, there's a medical answer. But for practical purposes, I, of course, consider it as a disease. We should be targeting it because, and that comes to the point that was made, that these interventions have to be super safe. But I once gave a talk, and a member in the audience came to me and said, "I'm taking this treatment XYZ," and I rolled my eyes because I thought, oh, boy, I wouldn't take it, you know. but then the person said, "You know, Steve, I'm now 85. If I do nothing, I run a high risk, you know." And I think we need to recognize it. If we do nothing and interpret aging as a natural process, we really let down many people who are counting on the biomedical industry to give better answers than simply exercise and eat vegetables. You know, we simply need to give people additional options.
We have lots of treatments that are used before people are actually diseased as prevention. It's okay. This fits with that if it works.
Right.
Polina, were you about to say something? You're leaning forward. I don't know if that's just
yeah. So I just want to say I don't see the issue with the classification yet. So I think that, I mean, there are companies out there like Unity Biotechnology, Restore Bio, and others who are running clinical trials, and they just don't use aging as an indication. What I think we can do as a community, we can participate in those trials. Of course, I don't think it's necessary, and I think it will be actually damaging to the trial if we will report the outcome on the biological introduction for clocks, because if, for example, if something that affects the body only locally and only against one particular indication, I think for FDA it will be red flag. It will show you also other systemic effect on the blood level and so on. At the same time, we can do this like in a double line way and still run it. I mean, also in terms of classification, the last ICD 11, it was just accepted. So the next one will be revised in 2000. Yeah, I think it's 2028 or something. So we still have plenty of time before we can change something in terms of classification. But I don't see it's such a huge problem because again, there are companies out there, they're running clinical trials on purely anti-aging compounds without having aging as an indication. And again, we can participate in those trials.
I don't think FDA would ever object to using other analyses to form new hypotheses in a trial. It doesn't damage the trial. You're not going to believe it necessarily because it wasn't prospective, but it's a good way to make new discoveries and think about things. That's okay. It's good.
Well, a little.
Okay.
So go ahead.
I just want to kind of reemphasize again what Luigi pointed out is that we should not just rely on the biomarkers we have, but use these clinical trials to design better biomarkers. So if there is something like TAME or a similar trial that actually shows a reduction in multimorbidity, we can, if we actually are collecting the data, we can go and say what things changed in accordance with that change in multimorbidity.
Right.
And from there, develop better biomarkers that we can move to other clinical trials for validation, rather than assuming what we have that's mostly been from cross-sectional analyses are the best that we're going to end up with.
Bob,
absolutely, that's how you learn.
Is it correct to think that the basic standard of evidence, substantial evidence, is going to apply here, so that it's great if someone comes up with a neat trial for a multimodal treatment or effect on aging on the elderly, but if we can show mechanism, causation, variability, validity, all those elements, those are things that the FDA would consider probative. I guess at the end of the day, you have the gold standard of the actual clinical validations, but as you mentioned in a planning session, there are ways to use the accelerated approval process to move the pre-approval, post-approval bar closer, so that the industry can make progress in developing their interventions.
But remember, accelerated approval is directed at particular very bad outcomes.
Yes.
That's what it's for. And as I said before, remember, you're talking about in some sense, treating everybody, you really want to be pretty sure that it works.
Steve, I know.
I think,
Bob, you are right. We need to be careful because we don't want to treat every single 80 years old person. But I tend to agree and disagree with Steve. I think that accelerated aging is a good indication. I think that if we start thinking about, there is a group of individuals that show that, the rate of accumulation of disease and functional decline is higher than the general population. I think that condition fit our, definition of a disease. In the extreme limit, we have the progerias. But between normal aging and progeria, there are intermediate events or conditions that we have not targeted that need to be addressed, at least at the beginning. To me, this should be our target.
But wouldn't you want to do a study in them? They're the perfect group to study.
Oh, absolutely. In fact,
yeah, well,
we're working exactly in that.
Then you have data. Then you're not speculating. You have actual data in that population. A very good question would be how much to broaden the indication. That's the sort of thing that have to be discussed.
I have a couple of comments. First of all, there is a high-risk group already, and these are people over, let's say, 80, By the way, once I turn 80, the high-risk group will be 85, but anyways. I mean, I think if there was a way of prioritizing, studies in people who do have a very high mortality risk, and everything obviously has to be voluntary, but it's my experience, some people are willing to try things, and so if there was a way to establish an infrastructure that allows very rigorous evaluations, that's what all of us love about clinical trials, right? You arrive at definitive answers, But if that could be developed, call it the 80+ interventional study, I think that would be valuable. I wanted to come to one other point, what really hampers the field of human biomarkers. And this is, we do not have a gold standard anti-aging intervention in humans, And you may recall that I showed you some nice results in mouse studies. Why? Because in mice, we do have gold standard anti-aging interventions, caloric restriction, for example, But so why do we need gold standard interventions? Coming to this point of causation and modifiability, we could say, well, this biomarker that was just published actually does not change in response to this gold standard intervention in humans. So therefore, maybe it isn't particularly useful. Let's stop.
We can go all day on this. Just a shout out, I guess, that Steve obviously feels very strongly about this. He's set up a foundation, the Epigenetic Clock Development Foundation, to explore ways to facilitate this conversation. This is really sort of a cultural exchange, a discussion between the geroscience researchers and the FDA. And this will undoubtedly be a continuing conversation for some years. And what I think we should do then is, and I apologize again for all the people who ask questions, we will try to have them addressed after the seminar. What I want to do here, I guess, is to thank the panelists in the audience for making this a terrific webinar. We will post the recording and the slides in a couple of days to a couple of websites that we'll let you know about. I think evoking an in real life conference, we'll keep the Zoom line open for a few minutes, and those panelists who can linger a bit, please stay, step off the virtual dais. And we can try to address a few more questions, whether typed in or perhaps we can try to do a call out. But let me release people formally from this webinar. Thank everybody, and then again, we're going to keep the Zoom lines open, and if folks want to speak up, panelists, Bob, even kind of open mic, we'll give it a try. Maybe you could put your name in your chat, in the chat function, saying you'd like to ask a question or speak, and we can recognize you. So thanks, everyone.
Thank you.
Can the panelists come off mute so we can at least speak? And well, what do you think? What have we been able to accomplish, scratching the surface, I guess, for starting this topic? What's clear is that it's got a lot of interest. There's several hundred participants, attendees, and others will be checking out the recording. And I don't know whether you have any sort of take-homes from this for whether it's altered your thinking or given you suggestions about lines of inquiry. Can we just have an informal chat? Speak up, whoever wants to comment.
There were so many wonderful questions in the chat session, and Thomas or somebody, if you could read some of them, we could perhaps answer them, because people
Sure,
went through effort of typing it in. Yeah. Mm-hmm.
Absolutely, we can start with that. Well, first one I see here, Steve, does your new foundation have a website? So obviously we'll post something, follow-up information when that's available, but let's see. So some questions, I think Carl Fleger had a question for Bob. I'd like to hear more details about what the bar is for getting a biomarker established as an approved surrogate endpoint. In particular, historically establishing has involved uncertainty about whether hypothesized mechanisms would have the predictive value. So we talked about this a bit, but Bob, has anything altered your thinking or amended, added to your thinking based on the fact that we have this webinar?
Well, I would say probably not. As I tried to explain, there are many plausible biomarkers / potential surrogates that haven't worked out because we didn't really understand all the things the intervention did. And I think any prospective biomarker has to be validated by using it in a trial. And I do think that it's possible to enrich the trial by picking patients in a prospective screening, by picking the patients who have a big biomarker response. That's a good way to do it. I also think you probably want to take people who are at higher risk, maybe older, for having the event. But those trials are possible. They've been done in many settings, and they can be done now. And while you're doing a focus on the primary endpoint, there's no reason not to look at other interesting endpoints, skin tone and all that other kind of stuff, which is sort of what the TAME trial is doing, if it ever gets if it ever fully gets off the ground. But
Right.
I didn't hear anything that made me think we don't need to have actual evidence that the biomarker predicts an outcome. Like all trials, you got to. Sorry.
This is Carl who asked the question. Thomas didn't read the last part of the question. What are your thoughts on machine learning and fancy statistical techniques that flip around the mechanism aspect versus the predictive aspect, right? Historically, we had mechanism, we thought it was predictive, but sometimes it turned out it wasn't. We needed the trial to validate it. The new world of strong AI is flipping this around. We have predictive things that we know are validated for their predictive ability, but we don't necessarily know the mechanism. How does that change your thinking?
Well, I'm not positive that I fully understand that, but if you think you have a marker that predicts outcome, and you think that changing that marker will alter what the outcome is, that's what you have to show. And I don't think your explanation of the mechanism or anything like that alters that fundamental requirement. You have to know that it really does that. And if and if it's a big effect, that won't be very hard.
I think that's absolutely true. I think that when you use a biomarker for clinical purposes, if that works, and to identify the people that need the treatment or track the effectiveness of the treatment or associate is a very good surrogate outcomes. Whatever it is, in some way we don't really care. other uses of biomarkers that allow us to understand the biological mechanism of aging and identify new targets for intervention, those really require understanding mechanisms. But I think that the two use of biomarker, it's complementary and but separated in a way. You know, even if we don't understand what it is, as long as it works, I don't think there's any problem.
You can use the biomarker to help you pick the dose, which is not a trivial matter. It can also, if you believe it, help you identify who the responders are going to be. That's the slide I referred to before. It was pretty striking that the intervention had a huge difference in effects. Well, for the people who don't respond, don't put them in the study, for goodness sakes. You're wasting everybody's time. Why not pick the people, if you believe in the biomarker and believe in the surrogate, it's a potential surrogate, pick the people who seem to respond by screening and then randomizing. Those are all enormous efficiencies, and they can make a trial much smaller. Look, you know, some of the early blood pressure trials had just a few hundred people in them. It's not that hard if the drug works.
So this is something we're going to talk about this at Modifiability as well, but who would perform these kinds of trials? You could have NIA, you could have, pharma is not really into the anti-aging or the longevity space yet because they're looking for a clear regulatory pathway and other things. But are these things that startup companies would look into? As was mentioned, if you have, Polina, I think it was, if you have a Restore Bio that's already looking at what I've kind of been calling neoclassical endpoints, those that are clinically meaningful, like enhancing immune response of the elderly. There's an interesting company in Texas that's for sarcopenia and skeletal-muscular purposes, where indications where they're measuring reduction in rehospitalizations. Those are clinically beneficial things. So you can certainly look at them. You don't have to look to see if is it generally rejuvenating biological aging. Where would those trials come from? Steve, are you thinking they would be?
Well, it's always worth thinking of the VA, although you have a no women problem.
Yes.
But it's an elderly population.
Yeah, I mean, unfortunately, the NIA can probably cannot afford these kinds of studies, you know, it's really there's really no other way than to involve industry, you know, to help finance it. But I think we already mentioned this tremendous opportunity really for the biotech to add these promising biomarkers, you know. Why? Because you can use your infrastructure that investigates a totally different treatment, you know, to make major discoveries. I think that's phase one. And what the field really needs is a success story, you know. We need one pill that truly extends health span by 5 years. That's what we need. Once this pill has been discovered, there will be a gold rush, you know, and we will never worry about funding again, you know, but that's what we need, you know. Conversely, if we have a big disappointment, if tens of millions are being wasted on a treatment that fails, then we will hamper the development of this whole industry.
It's a bit ironic considering the billions have been spent on certain other large indications by big pharma, whereas aging is the biggest of them all, it seems to me. But this is sort of open mic. I see that we still have a number of folks still with us. If you have any thoughts or questions you'd like to share or pose to the panel members and to Bob, especially to Bob, please mention something in the chat room and we'll recognize you. I got to get to the bottom of the chat, I guess. Sorry. Well, people aren't holding up their hands, which is kind of interesting to me. I would have thought that with the people who are left in the room, they would have questions. We still have over 100 folks. Why don't I just start going down the list of questions then? I don't want to make this overlong, but just pulling out something here. This is a question for Polina. How much more data do you need to achieve high performance in the models that you're looking at? You obviously are pretty confident about them, and they're getting better every day. But is there something you can say in response to that question?
Yeah, I think in terms of the number of samples, we're not that limited. We're actually limited by the number of data points available for the different data points available for the same individual. So if we want to actually connect those aging clocks, because as I said, there are many of those, and there is low or little correlation between them, we will have to design a study when we analyze them all at the same time, and that has to be a lot of data, but not in terms of number of samples. But in terms of number of samples, I believe, at least for blood biochemistry, we kind of reach the maximum error rate. I mean, the minimum error rate there possible, the maximum accuracy. So no matter what we're doing, and we right now are trained like on millions of samples, nothing is actually changing in terms of accuracy. So unless we have a completely new algorithm that help us, accuracy will not change. But yeah, we need more we need other data types. So not only single data type, but other at the same time.
I'm having trouble navigating the chat here. So Bronte, have you been keeping track of the questions? If you can pull out a couple and just read it out, we can have the appropriate panel member address it, please.
Okay. So some earlier questions that were asked include Mario asked, how do you control the multi-collinearity mixed effects in your deep longevity learning models during extrapolation? And that was directed at Polina.
Right. Yeah, we just do like a standard test to exclude the models that, exclude the features that are highly correlated with each other, because otherwise for deep neural networks, even though they're not like simple linear models, it will take a lot of time for them to converge. Probably they will fail to converge at some point. So we just do basic filtering. So no magic in that.
Okay. Denisa asked, metabolomics studies, can someone comment on what they see ahead in this area?
Maybe Steve or Luigi or...
Yeah. There were several publications that describe metabolomic aging biomarkers. I mean, these look very promising, So clearly you can estimate chronological age, to some extent. I'll make a comment that doesn't just pertain to metabolomics, but also proteomics, where there were very exciting biomarkers. And you can think of any omics, to an audience member, you may get frustrated because you hear about all these biomarkers, and every week there's a new, very exciting publication. So your head may be spinning. And however, actually the opposite is the case. We do not have enough validated biomarkers, and that comes back to what Morgan Levine talked about, and many of us who are statisticians talk about. Reliability is key. The so-called retest variability has to be low. Above all, the claims have to be validated by many independent groups, And so although your head may be spinning about all these omics that you are supposedly have to measure, and these could be expensive studies, there's really a dearth of biomarkers that have been validated, And to their credit, many people are working on that, But, and I think all of us agree that there will not be a single biomarker that measures aging, But the great hope is that we can develop 5, or let's say less than 10 biomarkers, that should be measured in every single anti-aging study. So in certain ways, if you don't measure these 10, let's make it 5. If you don't measure these 5 biomarkers of aging, you were negligent, in your study protocol, And this doesn't, it's not meant to be a restriction, because the more you measure, the better, but it would be a consensus view, and but we are still actually, well, I would say we already have 5 biomarkers that should be measured, at least in my mind, we have that, and, but yeah, I'll stop with that.
I just want to support completely what Steve was saying, and add, that, we were talking before about biomarkers that are really more for clinical use and other that are more for biological discovery. I think that one of the advantages of the protein and metabolomic biomarkers of aging is that they're closer to be effectors. In the pathways from gene code and, expression and then translation, protein and metabolites are there. And so we are, at least at this stage, we see traces of the underlying biology more in this biomarker than in others. For example, in the proteomics, when you do the proteomics that predict the chronological age and more recently change in health during age, you find that 50% of them are SASP proteins. So they are proteins that are secreted by senescent cells. And so that makes you think that there is an underlying mechanism that produce this biomarker and is not surprisingly associated with bad outcomes. So I think that that's an advantage, but I agree that we need that many of them, and as Polina is doing, we need to integrate them. We have not probably done, the work, enough work to integrate them, because each one of them can contribute to make our estimate more precise and may have a different use.
Last week there was a virtual conference, Aging Research and Drug Delivery, or Drug Development, and they had a neat feature on the Slack function where you could upvote questions, so you knew more people were interested in the question. One that's gotten one upvote here, and maybe we'll end with this. A couple people have asked, what are the 5 biomarkers you think we should be measuring now, so that everyone can go home having been guided by the current state of expertise?
You need to ask Steve.
No. I would let me ask Luigi. He's really, I call him the godfather of human biomarkers. He is the man who should answer that. And also Morgan, obviously, and the other panel members. Luigi.
But my answer would be not typical. I would say that I will not want to see any study on aging that doesn't have walking speed in it. Because that is so powerful, simple, that if we ignore function, is the strongest predictor of quality of life and the strongest predictor of mortality that we know about, the strongest predictor of disability. Of course, only in older people will work. It will not work in younger people. So it will not be sufficient. I think that the epigenetic clock that Steve have created really made the story of biomarker believable. Perhaps for the first time. Some of the audience is too young to remember that in the 90s, NIA made an enormous investment in a program of biomarkers, and it was a complete failure because the technology was not, ready for doing this. And so I remember that when we started talking about market, about aging, in the aging institute, people were, suddenly had to go somewhere. They had to, to make a phone call. Everybody was afraid of investing in biomarkers. But this time, we see the light. So that I think proteomics is going to give us a lot. And, some of the work that has already been done is good. I think that the technology we're using is still not perfect. There was only a couple of technology that we use in plasma And we already see what are the limitations. We need to be able to use multiple technology, including mass spectrometry. And I think that when we do that, our proteomic biomarker will improve substantially. I think that the microbiome is starting to be incredibly promising, especially in the study of the neurological part of aging. And so I think that research should be looked really carefully, because what's coming out is still interesting. These are four. I leave the last one to Steve.
Steve, you're on mute.
I allow me to mention Morgan Levine. So she developed a so-called phenotypic estimator. It uses albumin level, C-reactive protein. And Morgan, can you help me out? What are the other parts of that phenotypic age estimator?
Glucose, alkaline phosphatase, creatinine. Oh man, you're putting me on the spot. I think, and then white blood cell count, lymphocyte percentage, red blood cell distribution with basic clinical biomarkers. A lot of probably are what are in.
Yeah, that's what I remember as well. But just so that you know, so we love these high-tech biomarkers, machine learning, all of that. But these very traditional measures, that can be cheaply assessed and reliably assessed, are very powerful predictors. So don't discard old established clinical biomarkers of liver function, kidney function, immune senescence. The one thing we didn't mention are markers of immune senescence, perhaps, something like flow cytometric measures of naive T cells, naive cytotoxic T cells, possibly T cell proliferation tests, and especially in the day and age of COVID, we all start to appreciate how important these biomarkers can be.
There was a comment or a question about Nir Barzilay's comment. We actually had a webinar in the spring with Nir and Joan Manick from Restore Bio on the importance of focusing on host response, immune response, and the fact that it's great to develop vaccines, but the elderly don't necessarily respond that well to vaccines. So it's a great general independent strategy for COVID and future pandemics. Polina, since you were kind of, we were focusing on basics, do you have any, and everybody had a chance to add their favorite biomarker, do you have something that you're particularly interested in from a computational perspective or model perspective?
I actually would agree that clinical blood tests and clinical blood markers will be the one that we should measure. They will cheap to measure. If we think, about enrolling in clinical trials in clinical setting, that's something we should go for. They're standard, and that's something why we are, and this is why we're working a lot with blood biochemistry. But just as I said, they're not markers of aging by itself. Of course, there are some of them that are highly correlated, like interleukin-6, as we just said yesterday in our rehearsal call. Yes, some of them are highly correlated, or glycated hemoglobin is also highly correlated with biological age. But still, I believe that we need a model that sits on top of them. But then again, because they are so standard to measure, this model is rather easy to interpret. So you know that if your drug is actually like decreasing glucose or increasing albumin, you're probably doing something great, and you'd expect people to be pretty younger. So yeah, I totally agree with the rest of the panelists on the list.
Okay.
Tom and others, unfortunately, I need to leave. Thank you for the wonderful.
Thank you so much, Luigi. Thank you so let's release everyone with, again, hearty thanks to the panelists and to the attendees, including those particularly who were hardy enough to stay with us so far, still like over 100 people or so. So that's really indicative of the interest in this space. So thanks very much. Have a great day and a great weekend. Bye-bye.
Bye-bye. Thank you.